people often think of aging as some kind of natural phenomenon that is, in principle, not amenable to medical intervention. And that's complete nonsense. Aging is simply a biological phenomenon that causes things to go wrong with us, and as such it's perfectly amenable in principle to medical intervention. And furthermore, in terms of whether that's a good idea or not, really what aging is simply the set of precursors, the set of early stages of all of the age-related diseases that we're very clear that we don't want to get. So really intervening in aging with medicine would be just preventative geriatrics, and as such, it's sort of ridiculous, it's sort of completely messed up to even think about the idea that defeating aging with medicine would be in some sense undesirable.
So do you think our concept of aging, if it's something that's not amenable to medical intervention, has changed dramatically? I mean, because things that people used to think were dying of natural causes, we now have cures for.
I don't think that's the same thing. I think people have changed their attitude to exactly what counts as dying of natural causes.
Yeah.
And even the things that we very much don't have cures for yet, but nevertheless we still think of as diseases, that's happened, I agree. But the concept of dying of old age still very much exists in people's minds. They think of dying of old age as what you want to have happen to you. In other words, what happens if you escape all the bad things. But that's obviously makes no sense. It really just comes down to terminology. All it really means is dying without having got any of the things that medics have got around to giving names to, which is very different. In terms of feasibility, I think the main reason why I feel that things have changed is because we have made so much progress over the past decade, especially in developing regenerative medicine. So stem cell therapies, tissue engineering, a bunch of other things that would not normally be thought of when one mentions the phrase regenerative medicine, but which still come under that rubric in the sense that they constitute restoring function by restoring structure at one level or another, at a molecular or cellular level. So we've come an enormous way. We've got an enormous way to go in order to get to the point where regenerative medicine can have a comprehensive impact on aging. But to my mind, we are definitely within striking distance of that now. And that's what's changed, because for sure there's no way in the world that it would be conceivable to have an intervention that actually cured aging in the sense of like a one-off treatment after which one would not be aging until one was in some way reinfected. It's not going to happen, because aging is this very intrinsic phenomenon in the human body. But the idea of something that constitutes essentially repair, preventative maintenance, that's what regenerative medicine really is, something that will be applied periodically to the problems of aging. That has become extremely realistic.
So, I mean, in a lot of ways, we are prolonging life in our society at the moment. Our life expectancy is going up, and obviously in the last century, curing infectious diseases and getting rid of cardiovascular disease. But in terms of prolonging health, do you think that's more of an agenda than prolonging life?
Oh, absolutely. And I think actually one of the ways in which this conversation tends to get confused in the public eye is precisely because journalists especially tend to find it easier to sell papers by sensationalizing all of this and talking very much about the consequences for longevity without emphasizing the fact that they are simply consequences. In other words, that actually what we're talking about all the time is simply postponing ill health and thereby, as a side effect, postponing death. The only real difference between medicine in the future, prospective medicines against aging, and every other type of medicine is a matter of degree. The extent to which we would extend life is much greater once we think about really comprehensive treatment of aging than any extension of life that we've achieved with medicine so far. But still, all medicine is about extending good health, postponing ill health, or restoring good health, and thereby, among other things, extending life.
So would you say that your approach is prevention better than cure? Like, are you looking to reverse or halt prevent the ageing process, or a combination of all the three?
Good question. The terminology is difficult because each of these ways of describing it has connotations in people's mind. I've tended to end up gravitating to the word defeat aging because I want to emphasize that we are talking about something really that can postpone the problem indefinitely, not just something half-hearted. But equally, I don't want to use words like cure that have other connotations. Like the word cure, I feel, does have this connotation of one-off treatment that might actually permanently do the job without having to be periodic for a while. I think that's a fair way of talking about it. Because yes, we are talking about prevention in the sense of treating people who aren't yet sick. However, we're talking about prevention in a sort of to a lesser extent than some people are talking about prevention when they talk about postponing aging. Because a lot of people will think about it in terms of supplements and vitamins and so on, which are, if you like, trying to tune up metabolism, trying to get our bodies to work better in a manner that doesn't lay down these lifelong accumulating types of molecular and cellular damage so fast as it naturally does, and thereby, of course, to postpone, modestly if you're lucky, the ill health of old age. That would be even more preventative, if you like. And that, to my mind, is an approach which, even to the extent that it works at all, definitely doesn't scale. And we're not going to get significant postponement of ill health that way, where, let's say, decades, we're not going to do that by anything other than this preventative maintenance approach, this treating the precursors of age-related ill health.
So in your book, Ending Aging, which unfortunately we don't have any copies of, we're not able to get them in time, but I would encourage you all to seek out.
It's on its way.
It's on its way. Pop back next week. You talk about this concept of the longevity escape velocity.
Yep.
So I was wondering if you could just kind of talk about that.
Yeah, so longevity escape velocity is a term that I came up with in, I think, 2004, something like that. And the idea I was trying to convey with that phrase is that preventative maintenance, in principle, can postpone ill health, the malfunction of any machine, whether it's the human body or whether it's a simple man-made machine like a car. It can postpone it indefinitely, but only if the maintenance is comprehensive enough. So let's talk about cars for a minute. So if you if you do the sort of preventative maintenance on a car that the law requires, then the car works reasonably well for maybe 15 years if you're lucky. But if you do substantially more comprehensive maintenance on it, then as we know, cars can last 50 years or even 100 years and work just as well as they did when they were first built. So, and we don't have any 200-year-old cars, but the only reason we don't is because cars were not invented 200 years. So there's a threshold of comprehensiveness that's required. Now, the thing is that when you have nearly but not quite reached that level of comprehensiveness for a given machine, you are still buying time. So supposing we had therapies that were good enough that they could postpone the ill health of old age by, let's say, 30 years. So what that would mean is you would be able to take people who were already 60, just for the sake of argument, okay, and fix them up well enough that they would become biologically 60 again eventually, but not until they were chronologically 90. Okay. Now, if you just carry on using the same therapies at that point, then you would hit diminishing returns. Because ultimately, the reason why when I say the therapies are not 100% comprehensive, what I mean is there's certain types of damage that they don't work on yet, and those types of damage, those types of molecular and cellular problems, are accumulating anyway, irrespective of the therapies, and therefore, eventually, on their own, even however well you're using the therapies, those types of difficult damage, so to speak, are going to kick you over the pathological threshold, so to speak. But the thing is, 30 years is a long time in technology, including biomedical technology. So the chances are that if someone who has had these therapies at age 60 comes in at the age of 90, being biologically 60 again, they're not going to get the same therapies anymore, because the therapies will have been improved in comprehensiveness, among other things. So they'll actually get more thoroughly rejuvenated the second time around, even though the problem of rejuvenating them is intrinsically a more difficult problem. That's only true if the therapies have been improved in comprehensiveness by a sufficient amount. Right? And so that sufficient amount is what I call longevity escape velocity, the minimum rate at which we need to improve the comprehensiveness of the therapies in order that we can indefinitely stay one step ahead of the problem, despite having not yet achieved 100% comprehensiveness in how the therapies actually work.
And do you think we'll see longevity escape velocity in our lifetime?
I certainly do. I think that actually the first generation of therapies that give even a couple of decades of postponement of age-related ill health will be sufficiently comprehensive to achieve longevity escape velocity. And furthermore, that once we get there, we will have done the hard part. Everything that needs to be done thereafter will be essentially minor refinements and extensions of what we've already done. So in other words, just variations on the same set of themes. And therefore, should not be very difficult to continue to develop. I don't think we're going to see any significant postponement of age-related ill health until we get to a reasonably comprehensive level of sophistication on all 7 of the major categories of damage that I talked about when I described the problem. But once we've got even a reasonable amount, what I call the first generation therapy, which I think we have at least a 50-50 chance of getting to within about 25 years from now, then I think we're done.
All right, so let's talk about those. Let's talk about some science. What do you think those first generation regenerative therapies will be or look like? I mean, are they going to be pills? Are they going to be gene therapy? What do you think that entry level first generation might be?
Okay, so there's 2 questions there. There's what the therapies will be and there's how they will be delivered.
Yes.
So delivery will, I think, be pretty much by the sorts of methods that we're seeing already. It'll be injections, various things. It'll be perhaps some surgery, replacing organs. So there will be a lot of pressure to phase that out as the things get more sophisticated. But what we're delivering is the more complicated question. So, as I say, I tend to describe the problem of this accumulating damage that eventually causes ill health as being subdivided into 7 sub-problems. And these are categories of damage, types of molecular and cellular alteration to the composition and structure of the human body that happen throughout life, even starting before we're born, and that eventually contribute to age-related health one way or another. And so there are three types of change that are at the cellular level and 4 at the molecular level. The cellular ones are, number 1, loss of cells simply dying and not being automatically replaced by the division and differentiation of other cells. And that is the easiest one to describe in terms of what therapy will do, because you've all heard of it already. It's called stem cell therapy. That's essentially what stem cell therapy is, always, whether it's for a specific acute condition or whether it's for the gradual chronic loss of cells that we see in certain aspects of aging, like Parkinson's disease, for example. It's just you put in new cells because the body's not doing it for you. Second type of cellular damage is having too many cells because cells are dividing when they shouldn't. And of course that's what cancer is. And there are various ways to defeat cancer that are, of course, under a great deal of investigation. I've always been very pessimistic about cancer. I've always felt that it's by far the hardest part of aging to fix, simply because it has natural selection on its side. And the approach that I've been propounding for combating cancer is very elaborate and sophisticated, and to be honest, a lot of it I don't think it can be implemented. But I think it's worth trying a lot of different things. Basically I say we ought to indiscriminately, comprehensively eliminate the possibility for cells to divide indefinitely. Basically by doing suppression or really profound suppression of telomere elongation, both telomerase and also the alternative fraction that is used by certain cancers, but not only in the cancer. You see, the thing that really makes it very, very hard to kill cancer cells is that you have to also avoid killing all the rest of our cells. And the thing about telomere elongation is that it doesn't kill any cells at all for a while. Essentially, if I could wave a magic wand and sort of completely eliminate your ability to make any kind of extension of your telomeres in any cells, okay, so for example with gene targeting to actually eliminate the gene, then your cancers would not kill you anymore, would be great, but actually you die of anemia and stuff like that, because you've got various tissues that need a little bit of telomerase to keep their stem cells going. The thing is, if you actually do the arithmetic based on current understanding of the frequency of cell division of the originating stem cells in any rapidly renewing tissue like the skin or the blood or the gut, then it's come down around 10 years that you take before the stem cells reach their half-life limit. So one could, in principle, if one could, well, first of all, one's got to actually develop a sophisticated enough clean and comprehensive enough gene targeting to be able to actually eliminate this telomerase in the first place. But secondly, one's then got to essentially solve the problem of the eliminating the immortality of the stem cells. But one doesn't, because one could simply replace the stem cells.
Yeah.
And so that's the proposal, basically, to replace the stem cell, to replenish the stem cell pools in these few rapidly renewing tissues that really need telomerase every 10 years. And of course, it's a tall order, but it might not be so tall as to be impossible within 25 years from now.
But very expensive.
Well, even that might not necessarily be true. I mean, you know, we're getting that. It sounds hard, but hey, it's very expensive getting sick. These therapies are going to pay for themselves very quickly, even if they are expensive to deliver. Anyway, so that's number two. Number three, the third of the three cellular ones, is also having too many cells, but in this case not because cells are dividing when they shouldn't, but rather cells are not dying when they should. And this is something that most people don't really think of, because, you know, are cells supposed to die? Turns out, yes, there are certain cases in the body where cells are supposed to die. Probably the most obvious one is the immune system, where cells proliferate like crazy to get rid of an infection, and then most of them have to die in order to get out of the way so that other cells can divide like crazy to combat the next infection. There are certain circumstances in which the dying part stops happening, and the immune system essentially fills up with cells that are not doing what we want them to do. And there are various other cases in aging where the same sort of thing happens. Anyway, so we need to fix that. And actually, one proposal for doing so is to use the immune system against itself to identify what cell surface markers show, distinguish those cells from others. Another approach is suicide gene therapy, to essentially get toxic genes into cells that only actually express their toxic protein when the cell itself is expressing other proteins that are diagnostic of the problem. And that's been used actually in terms of proof of concept very effectively in mouse studies, with one quite recently looking at senescent cells, which we call senescent because they exhibit the same sort of phenotype as replicatively senescent cells in culture. A mouse model was made that has an accelerated accumulation of those cells, and they crossed it into a background where they could just use tamoxifen to activate a drug-inducible suicide gene to get rid of those cells, and sure enough, the result was extremely therapeutic. That was just published about 6 months ago from the Pena group in Harvard. And got a lot of press, and I'm damn pleased it did, because it really put the problem of these accumulating death-resistant cells firmly on the map in a manner that wasn't true before. As well as showing, of course, that potentially suicide gene therapy or something like it could knock these cells off.
But using the immune system against itself sounds quite dangerous.
Well, quite. And, well, not necessarily, because, of course, it's not really using the immune system against itself. It's using the immune system against whatever type of cell happens to be one you want to get rid of. And, you know, that's more or less part of what the immune system does anyway.
Yeah, vaccination.
Right. So then, there's 4 molecular types of damage. The first one is mitochondrial mutations. So mitochondria, as many of you probably know, are this very important part of almost all cells that do the chemistry of breathing. They do the business of combining oxygen with nutrients so as to extract energy from those nutrients. And they have their own DNA. They're the only part of the cell that has its own DNA. They don't very much, but it's essential, and when it mutates, the mitochondria doesn't work anymore, and it rather appears that the accumulation of cells like that, of cells that have essentially had all that mitochondrial DNA get messed up, is important in aging. It's still controversial as to exactly how it's important. No one really has a handle on precisely how, but there's plenty of evidence that shows that it sort of must have a widespread effect. So about 25 years ago, well, long before I came along, someone suggested that we might be able to fix this problem by actually just putting copies of the mitochondrial DNA into the nuclear DNA. Of course, modified in such a way that it still works, even though the DNA is in one place. The reason this is not completely crazy is because there are these 13 proteins encoded in the mitochondrial DNA, but there are 1,000 or more proteins encoded in the nuclear DNA that encode mitochondrial proteins that are perfectly happily, naturally imported into the mitochondria.
Gene transfer it happens all the time in lots of different organisms.
Well, that's right. That's right.
Between the
Yeah. Now, it turns out that even if we could do the gene therapy part, which of course is the hard part of this, to get suitable copies into the nucleus, we still have to do a bunch of modifications to overcome the resistance of these proteins to being imported back into the mitochondria. That is generally believed to be actually the reason why evolution hasn't done all this for us. We completed the job of transferring all the mitochondrial DNA to the nucleus long ago. But especially over the past couple of years, there's been a couple of advances which appear to make that problem look solvable. So we think we can do it now, and that's one of the projects that we're working heavily on in our lab in Mountain View, in California. Some of you may be thinking, well, why hasn't he mentioned the nuclear DNA? What's so good about the nuclear DNA? And in particular, what's so good about putting the mitochondrial DNA into the nucleus anyway? Surely you can still mutate and cause problems. Answer is, well, yes, but only incredibly slowly. The mitochondrial DNA is really badly vulnerable. It's in the wrong place. It's right next to the source of the main DNA toxin, so to speak, namely free radicals created by the mitochondria as part of its normal operation. It's not protected so well. It hasn't got, the DNA is not wrapped around these proteins called histones that protect nuclear DNA. And various other things that make the rate of accumulation of mitochondrial mutations vastly greater than the nuclear DNA. So that's why it would be protected. Nevertheless, nuclear mutations happen. They cause cancer, of course. So surely, what about the mutations that happen in the nucleus but don't cause cancer? Well, I firmly believe that we don't need to worry about that. And the reason we don't need to worry is simply because of cancer. In other words, since cancer can kill us with just one cell going seriously out of control, the risk, or the requirement not to die of cancer before we have had offspring, has driven evolution to develop DNA repair and maintenance machinery in the nucleus that's so good that everything else is protected unnecessarily well. And this is something that is so controversial, no one really knows whether it's true, but there seems to be pretty persuasive data from mice that shows that it almost certainly is true. If you look at the rate of accumulation of nuclear DNA damage during adulthood in mice with specific assays that are nicely unbiased and so on, then the answer seems to be that the rate of accumulation is so low as to be undetectable. And of course, if there's no accumulation to speak of, then it can't contribute to anything age-related. So it's looking good. I mean, it's still an open question. We are actually funding a project that looks at one of the potential loopholes in exactly what I just said. So it remains to be seen, but it's looking good right now. Okay, so what are the other three types of molecular damage? Well, there's another one that's inside the cell, like mitochondrial mutations, which is just molecular garbage. So metabolism, metabolic processes make byproducts all the time that have no particular function. And of course those byproducts are in general broken down or excreted, as the case may be. But if those byproducts are very, very rare, so that they're only created at a very slow rate, then in many cases evolution hasn't really, in some sense, got around to bothering to figure out a way to either excrete or destroy the stuff. So instead the stuff is sequestered and just accumulates gradually in the cell. And
sounds like a familiar way of dealing with waste.
Right, exactly. Yes. So of course eventually there's a problem, rather like if you don't take the garbage out of your house for a month. The house doesn't work so well. And the only reason evolution hasn't taken the trouble to deal with this problem for these particular types of garbage is because the accumulation is slow enough that in the wild we'd all be dead anyway from predation and starvation and hypothermia and stuff. And it's only now that age-related diseases result from this. But some of the most important ones do. So cardiovascular disease is caused by precisely this problem, by oxidized derivatives of cholesterol that macrophages don't know how to process, getting into macrophages and essentially poisoning them so that they eventually stop working in the artery wall and turn into what are called foam cells. They're sort of undead macrophages that are the first histologically visible stage of atherosclerosis. If macrophages could process oxidized cholesterol, we would not get cardiovascular disease, simple as that. It's the same for macular degeneration, which is the major cause of blindness in the elderly. That's caused by the accumulation of certain derivatives of photoreceptor material. That again, in this case, the retinal pigmented epithelial cells can't process. So the idea that we've been pursuing over the past few years which seems to be going rather well, I must say, is that we might be able to find enzymes in other species that can break down these things. Specifically, we're looking at bacteria. The reason we're looking at bacteria is because that's been an extremely successful way to find genes and enzymes that can break down unusual substances.
Especially bacteria that live in really extreme environments.
That, yes. It's been a really good way of doing it for environmental decontamination. This is an idea that was put forward more than 50 years ago now, and it's turned into this enormously commercially successful discipline, not just academically successful, in which they basically go and find bacteria that can break down such and such a substance, let's say TNT or dioxins or whatever, and they just grow them up in the lab and stick them into the environment and the pollution goes away. It really works incredibly well. Essentially because these substances are energy rich. If a bacterium is exposed to this stuff, then there's selective pressure to evolve the ability to break it down so that it can live off it. Now, of course, our idea is not to identify bacteria that can break down oxidized cholesterol or whatever, and then inject those bacteria into the body so as to get rid of the stuff. That would probably have side effects. But so our idea instead is to look at the basis for capacity, to identify what genes and enzymes the bacteria have that allow them to break down such and such a substance, and then incorporate those genes and enzymes in one way or another into our own cells, just augmenting our catabolic function. And so this is going really well. So first of all, we've become very good at finding bacteria that can do what we would like them to do. Number two, we've become very good at finding the enzymes, finding how to do it. Step three is where we are right now, putting those enzymes into mammalian cells, in cell culture of course first, and getting them to work. And that involves quite a lot of tinkering because, first of all, of course, the mammalian cell is much more complicated than a bacterial cell, and therefore you've got to do targeting to the right compartment, the lysosome in general, things like that. But long and short of it is, we've got our first preliminary results are getting there. We've just actually a month or so ago published our first paper showing genuine protective effects in cell culture against the number one, public enemy number one among the oxidized cholesterol derivatives, the one that's the most abundant and most toxic, for a particular enzyme that we found a year or two ago, showing that cell viability is improved if this enzyme is being expressed and targeted to the right place. So yes, we're pretty happy about how that's going. Of course, a long way to go. We've definitely got to get all this working in live mice as well as in cell culture, and so then we can think about clinical trials. But we're pretty happy about how it's going. It's gonna, and the thing is, once it works, it's going to be just incomparably more effective against cardiovascular disease than anything we've got today, or indeed anything that's coming down the pipe. Because everything we've got at the moment is just tinkering around the treatment of the pathologies rather than actually getting rid of the underlying cause. And so, anything that comes under the heading of geriatric medicine, you've got to remember that it's really a losing battle, because ultimately you're treating something which is caused by something that's accumulating. In other words, the geriatrician's job is intrinsically getting harder as the person gets older, whatever happens. Okay, so that's number 5. They're both molecular, and the other thing that they have in common is they're both extracellular. These are things that happen outside the cell. First one is simply molecular garbage again, putting it outside the cell. So a typical example is amyloid in Alzheimer's disease, for example, the stuff that forms these things called senile plaques. And here we have this is what I would say is intrinsically the easiest of the 7 categories of things. And the reason it's the easiest is because the machinery that we have naturally to break things down outside the cell is much more primitive and restricted in its function than what we have inside the cell. So if you could just get this extracellular junk inside a cell, then it types pretty quickly. And that's exactly what people starting in the late 90s started to try and do. They took an Alzheimer's mouse model and they essentially vaccinated against amyloid. So as to get the immune system of the brain, specifically microglial cells, to engulf this stuff, to phagocytose it, and sure enough, it went away, and they were able to get pretty good evidence of improved cognitive function as a result. Now, of course, the mouse models of Alzheimer's are actually rather poor, and not many people really think that if you could do the same thing in humans, then it would be a complete cure for Alzheimer's disease. But I think that it's quite likely to be a component of an eventual panel of things that we do that will cure Alzheimer's disease. So I'm delighted to say that this mouse result spawned a clinical trial awfully quickly. In fact, a little bit too quickly. They had to actually abort the first one. But the long and short of it is now we're in phase 3. Well, I said we, there's nothing to do with SENS Foundation. The phase 3 trial going on. Everyone's quite hopeful. The phase 3 trial was actually initiated before the phase 2 trial was over, which hardly ever happens. It only happens if the phase 2 trial is going so well. So that's a pretty good deal. And
so it's hoping that we'd see that then, I mean, the end of a phase 3 trial would lead to licensing.
This is exactly right. Phase 3 is the last stage before regulatory approval in the US. Yeah, so everyone's pretty optimistic about that. Now there's extracellular garbage elsewhere. We have a project that we're funding actually at 2 places, one at Harvard and one in Texas at Baylor College, that are focused on a different type of amyloid made of different stuff, which accumulates in the heart and appears to be extremely important in terms of the cause of death of really old people, people who are over 100 or even over 110. It's called senile systemic amyloidosis, they die of, and it's caused by a protein called transthyretin. We're working on that. But it's essentially the same concept we're using. We're trying to activate the immune system against this stuff. And finally, last one is stiffening. So, our bodies, of course, are not just made of cells, but also stuff between cells, and the stuff between cells is not just garbage. There's this thing called the extracellular matrix, this lattice of proteins that hold our tissues together. And that matrix needs to be elastic, especially in certain cases. So the artery wall is probably, the major artery is probably the best example. Of course, they've got to handle the constant pulsation of blood pressure from the heartbeat. And as life goes on, the artery walls get stiffer. And that's bad for us, that's very bad for us. It causes hypertension. It's the main reason why blood pressure goes up with aging, which of course has a massive number of downstream effects. So, question is, what's the stiffening happening? Why is this happening? Answer is, basically, it's almost all of it caused by a process called glycation, which is a chemical reaction, or rather a network of chemical reactions that starts with circulating sugar molecules attaching to proteins that are components of the extracellular matrix. Sometimes that sequence of reactions leads to chemical bonds between adjacent proteins, and the random distribution of those new chemical bonds is what causes the stiffening to happen. Now the good news is that the chemical structure of those bonds is totally bizarre. Very, very weird structures that happen as a result, completely different from anything that the body lays down on purpose. So that, of course, opens the door to the possibility of identifying drugs that can selectively break these unwanted crosslinks without messing up the extracellular matrix overall. And that's exactly what people are looking at. There was an initial breakthrough which we're not really sure still whether it really was a decisive one back in the 90s, looking at one particular, apparently very abundant type of crosslink. People didn't get very good results in clinical trials with that, and people think that's probably because that type of crosslink is less common in humans than it is in short-lived animals that the original work was done in. But it's now very clear what is the most abundant crosslink. It's a particularly weird one with a 7-membered ring in it. But anyway, we are actually engaged in a project right now to find ways to crack that one, to break it. And so yes, these are the things we're doing.
So you've got a lot. I mean, I just like to say, of all those exciting things that you're doing, what's possibly the one thing that stands out to you, or is there one thing that stands out to you as being like the next thing that's going to unlock sort of the next level, if you like?
Definitely, there is no one thing. No, we are the only criterion by which we prioritize one of these things over another, or within each of these strands, from some strand over another, is the extent to which other people are working on it already. We're quite small. Our budget this year is only 4 million or something like that. And so we have to think carefully all the time about how to get the best bang for a buck. And that means working on the things that other people are neglecting. That usually means the things that are at the earliest stage that people still don't quite believe they'll ever work.
That's challenging.
Yep, it's what a non-profit is for, really. You know, we're trying to get out of the short-termism that affects most people.
Yeah, and do you think that having a funding stream that's independent of traditional kind of grant cycles lets you do kind of more out-of-the-box blue sky research?
Oh, Christ, yes.
I mean, you mustn't be able to write the traditional sort of science grant to do
you're absolutely right. Essentially, it's easy to see why the private sector would not get into much of this. Simply, the time frames are a bit too long. And so they're into
fast track an aging experiment.
Well, actually, I'll come back to that because that's not certainly true. But the time frames simply for developing the technology, let alone testing them, are really too long. So the easier parts of all this, such as, for example, that trial to get rid of amyloid, those are very much attractive to the private sector. In fact, that trial is being coordinated by the company.
But only after the initial R&D is done by someone else.
That's right, yes. Now, you would think then, okay, not a problem, that's what publicly funded research is for, to do stuff that's too blue sky, to actually, attract the private sector. And that's true. Unfortunately, as those of you who are in science professionally will be acutely aware, there are plenty of problems with publicly funded science as well. There's not enough money for it, ever, and what that leads to, inevitably, is a massive bias against high-risk, high-gain work. It means that essentially almost everything that's funded is funded on the basis of feasibility. There's like an enormous penalty in the end to the scientists, and for that matter to the peer reviewers who approve the science, if things basically, if you fail, if you have a grant, and you get some money from the government and the experiment that you want to do does not lead to some high-profile publication, then you're screwed for the next decade. So people just work on stuff that they know is going to work. In fact, most of the time they work on stuff they've already done.
So you do get this incremental increase in knowledge rather than any kind of real...
Right, it's very, very hard to do radically high-risk, high-gain stuff. And so, yeah, that's exactly why we work in this. This means that our funding is overwhelmingly from philanthropy. It comes from people who believe that this stuff is worth doing and who don't have a constituency or anything like that to worry about in terms of keeping on side. So they can make their own judgment as to whether this is worth doing.
Great, so let's throw this open for questions. I'm sure there's plenty of people in the room who'd like to ask you something. Up the back, yeah?
Just on cancer, what prospect do you see for approaching the problem just from an early detection point of view, say using nanotechnology or something similar? Presumably, if we can detect cancer at very early stages, they're all curable.
Okay, so the question, I'm going to repeat the question if I remember to, because I know that people from the back may not be as audible as I am. The question about cancer, will we be able to bring cancer really under control simply, or at least predominantly, just by getting much better at detecting cancers really early? Now, I absolutely do want to say that early detection has enormous value, for sure and certain. The great thing about human beings is that we more or less never die of cancer unless and until a cancer gets to a point called metastasis, where cells start falling off the edge of the cancer into the circulation in sufficient numbers that very occasionally that cell will be able to re-engraft, so to speak, will be able to get a foothold somewhere else in the body and start growing again. It's then that cancers become pretty much incurable. And if we can catch a cancer well before then and get it out, then result. That's going to make an enormous difference. However, the problem is that cancer is, like all of these other things I'm talking about, an age-related disease. In other words, its incidence goes up with age. If you look at the demographics, you can get a somewhat more confused answer, but we don't want to get distracted by that, because that's actually generally believed to be what's called a selection effect. In other words, you see a decline in the incidence of cancer relative to other things because there is heterogeneity in population with regard to how soon they get cancer, and so you end up being left with the people who just got lucky of being inherently more resistant to cancer than others. So let's forget that. In any individual, the risk of getting cancer goes up exponentially as they get older. So, even if you can find cancers as quickly as possible, you've still got to have a way of getting rid of them, and you've got to get rid of them more and more and more often, and eventually you're going to hit diminishing returns. So one way or another, we've got to find a way to preemptively eliminate the possibility that cancers can ever get big enough to kill us, to metastasize, in other words.
If I can play devil's advocate for a minute, the world's overpopulated already. Why should we all live longer?
So I would respectfully remind you of what I said at the beginning, namely that the longevity aspect of all of this are a side effect. The question is, are you telling me that medicine is a wonderful, great thing just so long as it doesn't work very well? You see, I mean, this is how you've got to think about it, right?
Yeah.
I have no idea whether we're going to have an overpopulation problem, or how we pay the pensions, or won't dictators live forever, or anything like that. In fact, well, I do have a pretty good idea. Actually, I know damn well we're going to be able to solve those problems perfectly easily. But even if you disagree with that, Why don't you start to tell me whether you seriously think that the risk of those problems is worth letting people carry on getting all the diseases and disabilities of old age?
So I'm sure a lot of people are thinking this. I would like to live longer. Besides from better diet, regular exercise, and looking both ways before crossing the street, what should I be doing now to sort of prepare for therapies which are coming in 25 years' time?
Oh, you should be giving me lots of money. So I mean, I am actually, unfortunately, the tragic thing is I'm not kidding. At the moment, there is genuinely pretty much nothing that people can do over and above everything that your mother taught you. You know, don't smoke, don't get overweight. That's about it, right? Unless you happen to be one of those unlucky people who are aging unusually rapidly in one or another way, like Ray Kurzweil, for example, came down with type 2 diabetes at age 30. Okay. Unless you're one of those people, there's no evidence at all that there's anything that we can do that can detectably postpone age-related ill health if you're already the sort of person who's going to get to 80 perfectly happily without much trouble. Therefore, the only thing that can be done is to increase the chances of these therapies coming along soon enough to take you to the next break.
So you wouldn't recommend something like calorie restriction or anything like that?
I'm pretty pessimistic about calorie restriction. I think that the evidence at this point, and certainly evolutionary theory as well, tells us very strongly that calorie restriction will postpone age-related ill health and therefore extend life much less effectively in long-lived species like humans than it has been repeatedly found to do in short-lived species like mice and rats. Essentially, the logic goes, the only reason why something so simple as calorie restriction actually can have this extraordinarily complex effect, namely postponing all of these disparate aspects of aging, is because it's programmed. We've got genes that respond to nutrient deprivation in this particular way, okay, that reprioritizes our metabolic network. So the question then is, why would it do that? Why would we have those genes? And the answer is because it's good for evolution, right? It's good for maximising our chance of procreating. But then we have to take into account the fact that long famines are much rarer than short famines, and therefore that the evolutionary pressure to have machinery to survive 20 years just doesn't exist.
Okay. But what about, for example, periodic fasting or something of that nature, where there might be, would have had periods of food and no food, food and no food?
Yeah, I mean, the same applies. Essentially, that periodic aspect is, it's certainly true, it happens, and it's certainly found that intermittent fasting works at least as well as regular calorie restriction in many experiments that have been done. But still, the question is, what's the period? You know, like I said, it's still the case that long famines just don't happen enough, and therefore, it's rather like the fact that we lost the ability to make vitamin C. Something many of you probably don't know is that almost all mammals can make their own vitamin C. It's not a vitamin for them. There's only 3 times in the history of mammalian evolution where that capacity has been lost. It's been lost in apes, including us. It's been lost in fruit bats, and it's been lost in guinea pigs. And the reason is presumed to be, in all cases, that there was a period during evolution when the availability of vitamin C in the diet was so good, so reliably, that there was no pressure to maintain the genes. So these genes obviously got mutated, as all genes always do all the time in the germline. And of course, normally the reason we keep genes is because of selection against the mutations. In this case, there was no selection because the vitamin C was coming in the diet, and by the time the reliability became less, and the availability of vitamin C became less reliable, it was too late. So it's the same deal. We started off living less time and having more elaborate, more impressive metabolic acrobatics to postpone aging in response to famine. And as we started living longer in the ad lib scenario, where we have as much food as we like, so the evolutionary pressure to maintain that response to famine diminished, and we ended up with weaker machinery.
Sorry, just finishing off. Machinery, nonetheless, though. There's still some remnant machinery.
This is not known. This is my belief. I would say that yes, we almost certainly do still have the intrinsic, evolutionarily conserved ability to respond to nutrient deprivation with various changes to gene expression that clean up, that alter our metabolic priorities. It's just the alteration is much milder than it is in other.
Yeah, in theory, is there a limit up to what age you can extend life? I mean, is there a limitation? In theory,
is there an inherent limit to how long people can live? People ask this question a lot, but it's really a question that's founded on an internal contradiction, because it sort of assumes both that we have medicine and that we don't. That I believe that there is an inherent limit in how long people can stay healthy and therefore how long they can live in the absence of medicine. In other words, basically, there are certain aspects of being alive that are non-negotiable. You've got to breathe, right? And breathing is fabulously bad for you because it creates free radicals, which are bad for you, and which cause a whole bunch, which essentially are a major contributor to all the types of damage I talked about earlier. So that is going to put an absolute lower limit on how on the rate of accumulation of these various types of damage, irrespective of the little tweaks that distinguish different people from each other. But that's all in the context of a situation where medicine doesn't exist. If we can go in and repair those various types of damage that are being caused by breathing and by other non-negotiable aspects of our metabolism, then any limit that there might inherently be no longer applies.
Yeah, I have a friend who has been reading excitedly about a study done recently. I'd like to know if you've heard of it and have any insight, where rats were given buckyballs in an oil.
Yeah, okay, I've got to be a little bit downbeat about this study. It was a very spectacular result on the face of it. And so the first red flag is that it was published in a very niche journal and that it came out of a group from Africa, which is not known for the quality of its science. But of course, some good things still happen that way, so that's not the end of the story. It just got people to look more closely at the paper than they might otherwise have done. And there's a couple of big problems with that paper. First of all, actually there are plenty of internal contradictions in it, like the average lifespan that's mentioned in the text is way different from the amount that's shown in the graphs. Things like that. But there's more to it. There's more problems than that. To me, actually, the biggest problem is that this study looked at three groups. They had these rats that they looked at controls, obviously. They looked at normal lifespan. They looked at rats that were fed these fullerenes, and they had rats that were fed olive oil. Okay, an olive oil-rich diet. Now, fullerenes, well, they are this mysterious, uncharacterized pixie dust. You can't really say anything. You can't really form any preconceptions about what they might do. They might just be magic, right? But olive oil is not magic. Olive oil is something that we understand pretty well. And they got at least half as much life extension from the olive oil as they did from the fullerenes. And that was still, off the scale, far longer than West Star rats are supposed to live. So there's a whole bunch of ways that the experiment could have gone wrong. They could have actually ended up, mistakenly using some bizarre species of Tunisian rat that actually naturally lives a long time, and not using West Stars at all. They could have, some technician could have replaced the rats. There was this famous story about replicative senescence, about the whole concept of telomere shortening causing cells to stop dividing, where Hayflick's predecessor as the mentor of all of this thing, a guy named Alexis Carrel, had come to the opposite conclusion because his technicians were keeping him happy by replacing the cells every so often. That's the honest truth, that's documented. Anyway, so long and short of it is, no, this paper currently has very low credibility in the community.
About the economics, you mentioned you've got a small budget to do some public research. Do you think as it gets closer to applicability, there'll be a scramble, a sort of patent thicket develop, and possibly the research slow down as private?
Yeah, excellent question. So people are very interested in what you might call the end game here. At the moment, a huge proportion of the research that needs to be done is at this early stage that I mentioned. I guess using business terminology, it would be called pre-competitive. And so there is, of course, the gradual accumulation of IP, of intellectual property, both in the private sector and in the public sector. We even have a couple of patents and we get some more that may lead to the spin-off, startup companies and so on. Personally, I'm not at all worried. And the reason I'm not is because people care so much about aging. Aging is going to be different in this way. If we think about today's situation, we've got this massive thing called the anti-aging industry, this huge industry that's based entirely on stuff that basically doesn't work. Right? Now, I think that tells us exactly how huge people's excitement is going to be for stuff that actually does work. So now, here's the way that I see this playing out. At some point in the relatively near future, less than 10 years I would say with high probability, we are going to reach a point in the lab with mice that is exciting enough that it's not just people like me, weird mavericks with beards, that are saying that this is going to change the face of aging. But people in general, acknowledged experts the world over, are going to be willing to go on camera and say, yes, it's only a matter of time before regenerative medicine for human beings becomes comprehensive enough to actually postpone aging indefinitely. Now, when that happens, that is when the real pandemonium is going to occur. Not when the therapies arrive, but when the therapies become widely anticipated. Because, of course, science is the new religion. Once the scientists say it, the opinions, Oprah Winfrey will say, let's actually do it sooner rather than later. And at this point, it will become impossible to get elected unless you have a manifesto commitment to have a war on aging, one way or another. Now, what does the war on aging actually mean? Of course, it means putting a humongous amount of money into the science. The science that we need to do right now in the lab, I would be running out of ideas on how to spend money if I had more than $100 million a year. If I had one order of magnitude more money than we actually do have, if we had 30 or 40 million, I'd still be pretty happy. But that's because it's all in mice. Once we get to the clinical trial stage, it's vastly more expensive. But that's fine if there's electoral pressure to do it. Right? So that's fine. Okay. But of course, not just the research. There's also all the tedious stuff, all of the investment in infrastructure, building the hospitals to provide this therapy when they come along, training enough medical personnel to provide them when they come along, and, exactly what you said, making sure that the intellectual property structures, ownership and incentives and so on in the private sector also are compatible with and conducive to the hastening of these therapies being available as quickly as possible. Another thing, actually, that comes into this, which I'll mention because it often comes up and it's closely related, is the mode of delivery of these therapies once they come along, where delivery means access. A lot of people say, oh dear, these are going to be terribly expensive therapies. Terribly expensive therapies today tend not to be terribly universally available. They tend to be only available to those who have the money to afford them. That would be awful, greater inequality, mumble. Now, of course, there's a generic answer that says, well, yes, that's true for all technologies. If we care enough about them, then we put more investment into lowering the price and everything becomes trickle down level. But there's a better answer, which is that not having these therapies, in other words, letting people get old, is itself extremely expensive. The only reason why therapies for the diseases of old age today are limited by those to pay is because they don't work very well. And therefore, you may postpone those diseases of old age by a tiny little bit, but then you've still got the outlay on people while they're sick after they've got these diseases and when they've got serious, that you would have had if you hadn't used the therapies that didn't have the small delay. If the delay can be indefinite, you never get that expense at the end. And there's a well-known statistic which says that throughout the industrialized world, the amount of money spent in medical care for people in the last year of their life is more than in the whole of the rest of their life put together, irrespective of how old they are when they die. Okay. So we're saving all that. Plus also, of course, there's all the indirect costs to do with, the people who would be sick actually are not sick, and so they're contributing wealth to society. Their kids are no longer having to look after them, so they're more productive, all those things, right? So the economic arithmetic is completely clear. We are actually about to start funding a study looking at this in much more detail, looking at everything you can imagine, economics, not just economics, but demographics and politics and everything about that, by a very well-respected group in Denver. But even without any of that study that we want to do, we can definitely say that it would be economically totally suicidal for any industrialized nation not to invest in this and thereby save the money that would be required.
We've got time for one more question. Yeah.
Has any of the research on existing organisms that exhibit negligible senescence been relevant to your work?
So the question is, does any of the work that's been done on organisms that already don't seem to age have any relevance to what we do? Unfortunately at this point, no. There are a number of organisms which either seem not to age at all, or which seem to have much better defences against aging than we do. But unfortunately, in almost all cases, the reason they do makes them irrelevant to us. And in the cases where it's relevant, the extent to which they do better than us is so mild that it's unlikely that we would be able to make much of a difference. So let me give an example or two. There are a number of lower animals, invertebrates, that seem really not to age at all. There's a famous one, some clam that was found in Iceland not long ago that's about 400 years old. There's an even more famous jellyfish type thing that seems to undergo actual reversal of development and go back to an embryonic state. It's called Turritopsis. Things like that. Problem with these things is that they don't have a central nervous system. At least not in the way that we do. Now, I don't know about you, but I'm quite fond of my central nervous system. The reason that's a big deal is because the central nervous system, and of course one or two other things like the heart, that function arises from being made up of cells that are designed to be non-dividing for the lifetime of the organism. And cell division is an extraordinarily powerful rejuvenating mechanism. It dilutes away non-genetic damage, it selects away genetic damage. It's a great thing. If you decide to create an organ, whether it's the brain or whatever, that doesn't do that anymore, then you are selling your soul to aging. Aging has become an enormously bigger problem. So we can't really use animals like that. Then there are animals like, there are certain types of fish that live an awfully long time, certain types of amphibians that live a pretty damn long time. But unfortunately, in general, those animals tend to be ones that grow throughout life, even throughout adulthood. They have what's called indeterminate growth. And that, again, is a big deal, because that means that every single organ in those organisms has the capacity for adding cells, for new cell division and cell incorporation. That means that cells can die and be replaced because the system for putting new cells in is there. If you've got fixed size cells like you and me, then organs don't grow during adulthood, and therefore there's no evolutionary pressure to maintain the ability for those cells to be replaced when they die, because the alternative exists simply to keep the cells alive in a non-dividing state. And in evolution's case, where you don't have to worry about aging because we're dying of predation and so on, that's not a problem. Going for a non-dividing cell that lasts a long time is easier. So again, we've got a problem because in the brain, for example, it would be fine if we had slow but steady neurogenesis, replacement of cells every so often throughout the brain, but unfortunately we don't. We do have it in one or two parts of the brain, but not in the whole brain. So yeah, these are the sorts of reasons. One good example where we might be able to learn something is there are some mammals, in particular the mammal called the naked mole rat, which not only seem to live a lot longer than closely related animals that are the same sort of size and so on, but also they don't get cancer. They really don't get cancer. Never been seen in a mole rat. And nobody has the faintest idea why that should be. Well, that's not completely true. There were some recent discoveries showing additional defences over and above what we've seen in other animals, but they seem to be, well, we're pretty sure that there's a lot more still to be discovered about how they do it. I think we might be able to learn something there, but that's pretty much the only case where I think we can learn something from animals that do impressively well in terms of aging better than us.
I'm afraid we're going to have to leave it there, but thank you very much, Aubrey de Grey.
Thank you.